Right, welcome back everyone. I hope you had a good break. The second session's work that we're going to look at is that of the DNA profiling or DNA fingerprinting, as well as genetic engineering with a bit of cloning. So let's start with the DNA fingerprinting. Okay, DNA fingerprinting nowadays, most people prefer to use the word DNA profiling or the term DNA profiling, okay? But it is the same thing. So that's, I think, the first thing that's important to know. If you come across either of those terms in a test or an exam, it actually refers to the same thing. The reason why most people prefer to just use profiling, okay, in this instance, is that fingerprinting is something that was or has been around for very many years. In the 1930s, we realized that people have individual fingerprints. Okay, so it could be used in crime scene investigations, if people burgled a place or broke into something and they touched surfaces, then we could actually lift those fingerprints, okay? I do have a picture here which I can just show you. Okay, so ordinary fingerprints are going to look like this. They are specific to individuals. No two people have the same fingerprints, okay? It's just like your DNA, and we're putting identical twins aside just for this section of work. But unless you're an identical twin, nobody else is going to have the same fingerprint as you. Now, this is just often where the confusion comes in. When people speak about fingerprinting, then we start to kind of think of the old fingerprints. So DNA profiling is just a term that we use in place of this to limit the confusion that we have. Okay, so what we have to look at is, we have to look at what is a DNA profile. Okay, that is the first thing that we have to look at, and the first thing that you have to be able to identify, or the first thing you have to be able to explain. So a DNA profile shows us the patterns made by an individual's DNA when we push it through a biotechnical process or biotechnological process. Okay, we take a person's DNA, we remove it from a nucleus. Okay, what you actually do is you make copies of that DNA, okay, which we can do with restriction enzymes and things like that, and then we push this DNA through actually agar gel, and there are electrodes, okay, and as the adenine and the thymine and the guanine and the cytosine, which is term one's work, okay, far back, as those nitrogenous bases move through the agar gel from the one electrode to the other, they make little patterns, okay, that kind of look like this. You might have a collection of them there, then there might be a big space, then you can have a little bit more, then you might have a very thick bar over here, an accumulation, okay. So as these nitrogenous bases get stuck in the agar gel, it makes a DNA profile. Okay, now like fingerprints, normal fingerprints, okay, are unique to individuals, so is a person's DNA. And what we're going to see is that the patterns that these bars make are going to be used to set up a profile for a person or an individual. So if you look over here, okay, it's just drawn a lot more neatly than I can. Here you have different patterns that have been taken, obviously, DNA samples from different people, and this is what your DNA profile is going to look like. Okay, if you want to have a look at an actual DNA profile, if you look very, very carefully, okay, over here, here you have a man who is studying the DNA profile of people or of an individual, okay, and probably in relation to other suspects or other individuals, okay, but you can really see that on this paper, you actually have these specialized and specific, rather, patterns that the DNA makes as it's being pushed through. They take a photograph of that, and now you actually have a lot of tangible information in front of you. Okay, you're able to look at these patterns and then decide in relation to another DNA sample possibly that you have. You're able to compare them and make deductions from there. Okay, so these are the things, if you are asked, come test an exam time, they'll probably be asking it in this way, but if it's practical work, it can be asked in a slightly different way, which will be based on a picture that looks more like this, and I will do an example with you in a moment. Okay, so a DNA profile is taking the DNA of an individual, pushing it through and using biotechnology, okay, in this process, and getting set patterns, okay, of what a person's DNA looks like. These are very specific to individuals. Okay, individuals have their own pattern, which is unique to them. Okay, so your DNA, like your fingerprints, your DNA is specific to you. It is the thing that makes you unique genetically, okay? So we are able to, by making a DNA profile or by looking at a DNA profile of a person, we are actually able to see what their genetic code is, and we see this by the patterns that their DNA makes when we push it through a process and we create a DNA profile. Okay, so what we have to look at, this is a very useful thing that we can have, okay, because there are a few uses of DNA profiling. Okay, in no particular order, but one of the first ones that we can look at is that we can actually diagnose inherited disorders. Okay, that's the first thing that we can use DNA profiling for. If we diagnose, okay, inherited disorders, we're looking at things like hemophilia or even sickle cell anemia. Okay, because these are inherited disorders. Inherited means that it's passed on from parent to offspring. Okay, it's something that you're actually going to be given in your genotype. Okay, hemophilia we know is a sex-linked disorder. Okay, so it's going to affect males more than it does females. Sickle cell anemia is not sex-linked, but it is inherited. Okay, so there's a family history of it. So DNA profiling is able to look at the DNA profile of organisms or individuals. We are actually able to isolate genes on specific chromosomes, and then based on that we are able to see, do you code, or do you rather carry the code for things like hemophilia or for sickle cell anemia or any other list of diseases that we've studied and that you have. So diagnosis is something that can often help parents, okay, which links to genetic counseling. It can help parents either plan to have children and know that there are risks involved if they have boys or girls in this case. Okay, sickle cell anemia we said is not sex-linked, but by using genetic counseling and looking at the parents' DNA profiling, we can work out what is the chance of having a child with something like this. The second thing which links to this is that we could possibly develop cures for inherited disorders. Okay, so that would be something that is also obviously up and coming. Some things have already had a lot of work done so far. It links to things like stem cell research, okay, which we'll speak about in a moment when we do cloning. But DNA profiling, if we are able to isolate genes and we're able to study those genes and we are able to really look at the DNA profiling of an individual and specifically at certain disorders, then we have a high probability of at least making some headway into looking for cures for these disorders. So this would be a definite advantage, okay, as the first one, to why we could be using DNA profiling. The third thing which we could use it for, which I think a lot of people kind of link this section of work to automatically, is that of biological evidence. Okay, so this would be like the police work and the forensic work. Okay, I'm going to look at an example with you in a moment. But here you would look at things like if blood was left behind at a crime scene, or semen, or hair samples, or skin, things like that. Anything that kind of has DNA that you can check in. Then we can use biological evidence to often help the police to solve crimes. Okay, so we can say, here is the sample, you might have 7 suspects, and if we look at, again, the patterns that are made, and you can, link it, or you can compare it to samples, you can actually use DNA profiling as biological evidence. It's something that has not only, come about in the last, while, it's really been around probably from the late 1980s, early 90s. As far as I remember, we've been using biological evidence in courtrooms and in police investigations. Okay, and then another thing that we could use DNA profiling for is for checking something like identifying relatives. Okay, so here would be things again like kidnapping. Okay, if babies have been taken from hospitals, you can now use DNA profiling to check if you do find the baby eventually, or find a missing baby, you can do a DNA profile to match it to the parents. Okay, so you could check the DNA from the mom and the dad. Okay, or if you only have the mom or only have the dad's DNA and you have the DNA from the baby, you are able then to check for relatives. Okay, sometimes in the cases of adoption, okay, this is something that can also be very useful. You can, if you have found out that you have been adopted, you can contact agencies and they can actually put you in touch after the age of 18 with your biological relatives. And what you could do is just to put your mind at ease and to make sure that it is genuine and that these are your family members. You can do a DNA profile and to see what the relation is between you and a possible brother. Sometimes it might be a half brother. Okay, that will still be able to be done, and you will be able to check that by using DNA profiling. Okay, so there are really good reasons as to what the value of DNA profiling is, what we can use it for. Okay.